Hi everyone. In this video, we're going to take a look at the process of replicating the leading strand of a DNA molecule. Before we get into the mechanism, let's just quickly recap what we mean by the leading and lagging strands. Remember that this is due to the anti-parallel nature of DNA and replication that proceeds outwards in both directions from the origin. Remember that DNA polymerase III, which will build the new DNA strands, can only work in the 5' to 3' direction. Now this is nice and easy for the sections where the replication fork is moving towards the 5' end of the template, since this will be the 3' end of the new strands. So these will make up our leading strands, which we'll look at in more detail in just a moment. The other sections will have the 5' end of the new strand moving towards the replication fork. And these will make up the lagging strands, and we'll look at those in more detail in another video. Here we are zoomed in to one end of our replication bubble, and we're going to be building the sequence complementary to that shown on the bottom strand here. We'll start by bringing in our primer, which remember is a small piece of RNA rather than DNA, usually around 10 nucleotides long. Once that's in place, we can bring in DNA polymerase III, which will progress down the template from the 3' to the 5' end, adding new nucleotides at the 3' end of the new strand. Now, as the fork continues to move, DNA polymerase will follow it until the end of the strand is reached. We'll look at the replication of the lagging strand in another video, and in that video we'll also look at the final steps required to finish the new DNA molecule. Thanks for watching, and please be sure to check out the other videos on DNA replication.